So guys, welcome back to our channel. This is Tom Waswa and I'm here to take you through biochemistry revision questions. So this is the first series of our revision questions. I'll be posting more and more of the questions. So kindly ensure that you like, share and subscribe to the channel so that you receive more and more of the questions. So this is just being the first series. I'll be posting more and more of the questions. That is due to public demand. So other than questions, I have also posted notes of biochemistry. So to get my work, search for Tom Waswa. Search for Tom Waswa on YouTube. Like, share and subscribe. I really appreciate. So we go to the first question. What are the major pathways of glucose metabolism? So what are the major pathways of glucose metabolism? So the first pathway of glucose metabolism is glycolysis. Glycolysis. The first pathway is glycolysis. So glyco means sugar. We can break the word glycolysis into two major parts, glyco and lysis. So glyco means sugar and lysis means breakdown. Lysis means breakdown. So glycolysis is a series of reactions that ultimately breaks down glucose into two pyruvate molecules, two water molecules, and also two NADHs. Then we go to the second part of the answer, gluconeogenesis. 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 So gluconeogenesis can be broken down into three major parts. We have gluco, we have neo, and also genesis. Gluco, neo, and genesis. So gluco means glucose. Gluco means glucose. Neo means new. And genesis means creation. Genesis means creation or synthesis. Creation or synthesis. So gluconeogenesis refers to the creation of glucose from new sources other than carbohydrates. Others say it is the creation of glucose from non-carbohydrate sources. So you need to ask yourself, what are these non-carbohydrate sources? So the non-carbohydrate sources include pyruvate, lactate, amino acids, and glycerol. So that is it. Then we go to another part of the answer is glycogen synthesis. Glycogen synthesis. This one is self-explanatory, refers to the creation of glycogen. Yeah, so this is the creation of glycogen from glucose monomers. Remember, glycogen is a polymer of glucose. Then we go to the other part of the answer is hexose monophosphate shunt or HMP shunt. So hexose monophosphate shunt refers to that pathway that oxidatively breaks down glucose and forms one of the major important components of glucose oxidation known as NADPH. This is a very, very important reducing agent in biochemical reactions. So we go to another part. We go to question number 2. Question number 2 says, explain the importance of phosphorylation of glucose during glycolysis. Explain the importance of phosphorylation of glucose during glycolysis. So we do phosphorylate glucose because of the following reasons. Reason number 1 is polarity. So a phosphate group is polar. So when we phosphorylate glucose, we make it more polar. so we make it more charged. So the importance of making our glucose molecule to be more polar is to trap it in glucose so that it does not easily exit the cell membrane. So the negatively charged phosphate group prevents glucose from moving out through the cell membrane. Then also structure destabilization. So when we add a negative moiety on the glucose molecule, it becomes more unstable, and this makes it more likely to undergo glycolysis. Then we go to question number 3. State and explain 4 major properties of amino acids. State and explain 4 major properties of amino acids. So the first property is optical isomerism. The first property is optical isomerism. So how do you define the term optical isomerism? So optical comes from the word optics, and optics means light. So an optically active isomer refers to that isomer that is capable of rotating plane polarized light, either to the left or to the right, leading to formation of 2 different isomers, L or D isomer. So what confers this property to amino acid? It is the presence of asymmetric carbon. The presence of asymmetric carbon. It is the presence of asymmetric carbon. So what is the meaning of asymmetric carbon? I think I have explained that one in my notes. But an asymmetric carbon refers to that carbon atom that is attached to 4 different groups of atoms. Let's say this is A, this is B, this is C, and this is D. So an asymmetric carbon refers to a carbon atom that is attached to 4 different groups of atoms. So that is A, B, C, and D. We go to another property, acid-base or charge properties. So a given group of amino acid have both acidic and basic properties, and therefore they are referred to as ampholytes. So at a neutral pH, an amino acids exist as follows. We have said that it has got 2 ionizable groups, that is, it has the amino group and a carboxyl group. I have also explained that one in my notes. So when the pH tends towards negative, towards neutral, that is, our carboxyl group tends to donate its proton to amino group. So this makes our amino group to be positively charged, while the carboxyl group will be negatively charged. So a substance that has got both basic and acidic property is referred to as amphoteric. Or an ampholyte. Then we go to another property, buffering action. So buffers refers to salts of weak acids. So amino acids are buffers. So they resist changes in pH when an acid or a base is added. So another important question that you need to have in your mind is why are their buffering action not significant in our bodies, yet they are buffers? Well, amino acids are simply weak acids, so their concentration in our bodies is insignificant, so their impact is low. Then ultraviolet light absorption. So under normal circumstances, amino acids do not absorb visible light, but aromatic amino acids such as tryptophan have got a capability to absorb visible light. So tryptophan can absorb visible light. So I have also explained the structure of a tryptophan in my notes. So you go through my notes that I have uploaded, search for Tom Waso, you'll get notes on the same. Then we go to question number 4. Describe Krebs cycle. So Krebs cycle also means TCA cycle or tricarboxylic acid cycle. So let's say you have an exam on Monday and you are reading the citric acid cycle on Friday. How do you easily remember those molecules? Well, I'm here for that. So to describe the TCA cycle, you begin by naming the major molecules of the TCA cycle, the major compounds that are involved in the tricarboxylic acid cycle. So I came up with a mnemonic on the same. That is the easiest way to remember. So I said original citrate is a successful, strong, furious molecule. Original citrate is a successful, strong, furious molecule. So let's begin. Original. Original means oxaloacetate. Oxaloacetate. Let me delete this. Original means oxaloacetate. Then this citrate means citrate, just as it is. Citrate is citrate. Is for isocitrate. It means alpha ketoglutarate. Alpha ketoglutarate. You know the spelling of alpha ketoglutarate. I'm just writing as a short form. Then successful, succinyl. Strong, succinate. Furious, fumarate. Then molecule, malate. Molecule means malate. So that is how you can easily remember those major molecules. Original citrate is a successful, strong, furious molecule. So original stands for oxaloacetate, citrate is citrate, is isocitrate, A is alpha ketoglutarate, successful, succinyl. When you look at the two words, they rhyme. Strong means succinate, furious, fumarate, then molecule is malate. Then I have another mnemonic on enzymes, but let's begin with the molecules first to draw the major skeleton of the citric acid cycle. So we can draw the major skeleton by doing this. So we say that O, original oxaloacetate. So we begin by writing oxaloacetate. Oxaloacetate. Original citrate. So the next molecule is citrate. Then we go to isocitrate. Isocitrate. Original citrate is A. So the next molecule is alpha ketoglutarate. Alpha ketoglutarate. So we have said original citrate is a successful. That means succinyl-CoA. Is a successful, strong. So that is succinate. Furious. That is a fumarate. Then molecule means malate. So by that you have drawn the major skeleton of the TCA cycle. Sorry for that interference. So you have drawn the major skeleton of the TCA cycle. Then you need also to remember the enzymes of the TCA cycle. So the mnemonic that I have come across with is so at Damascus, so at Damascus, so at Damascus, David slaughtered down 5 demons. So at Damascus, David slaughtered down 5 demons. 1, 2, 3, 4, 5, 6, 7, and 8. So with these enzymes, by this mnemonic, you can easily name the enzymes. So where we have an S stands for a synthase. Then the second S stands for synthetase. So just know that an S is for synthase. We shall see the difference between synthase and synthetase during our discussion. So we have where we have an S, you put there synthase or synthetase. Then at is aconitase. Damascus, this is dehydrogenase. This is dehydrogenase. Where we have a D, you put there dehydrogenase. S stands for synthetase. This is the second S, so this is a synthetase. Then D for dehydrogenase. F for fumarase. Then another D for dehydrogenase. Another D for dehydrogenase. So we can use this mnemonic to put our enzymes on the TCA cycle. So let's begin with the first one. It is a synthetase. What are we synthesizing? We are synthesizing citrate. So our enzyme is citrate synthase. Citrate synthase. Citrate synthase. We go to the next. The next enzyme is aconitase. The next enzyme is aconitase. Then followed by, this is a D. D stands for dehydrogenase. So what are we dehydrogenating? We are carrying out dehydrogenation on isocitrate. So our enzyme is isocitrate dehydrogenase. Then we go to the next step. This is another dehydrogenase. What are we dehydrogenating? What are we working upon? Alpha ketoglutarate. So alpha ketoglutarate dehydrogenase. We go to the next enzyme, S. S, we have said it is a synthetase. So what is it working upon? Our synthetase is working on succinyl-CoA. So succinyl-CoA synthetase. Succinyl-CoA synthetase. We can proceed. This is a dehydrogenase. So our substrate we are working upon is succinate. So succinate dehydrogenase. Succinate dehydrogenase. We go to the next step. We go to the next step. So our next enzyme is fumarase. That one is just the way it is. Our next enzyme is fumarase. Then we go to the next step. Hope you understand this, fumarate. So our next enzyme is fumarase. So our last enzyme is a dehydrogenase. What are we dehydrogenating? Malate. So our enzyme is malate dehydrogenase. Malate dehydrogenase. Then another thing that I said at the beginning of our cycle is the role of a dehydrogenase enzyme. So dehydrogenation, from the basic chemistry, you know that it is the transfer of a hydrogen from a molecule onto another. So where we have a dehydrogenase enzyme, probably we are moving a hydrogen atom from one molecule onto another. So we are through with this mnemonic. We have labeled our enzymes. So we want to see how to put the core enzymes on our cycle. So we've said that the role of a dehydrogenase enzyme is to facilitate the transfer of a hydride or a hydrogen from a molecule onto another. So when we say dehydrogenase enzyme, we are transferring a hydride or a hydrogen atom. So dehydrogenation normally involves the use of coenzymes. So the coenzymes that are involved are NAD and FAD. So let's see what is the difference. So dehydrogenation is in the step number. This is our first step. So first of all, let us label the steps so that we don't get confused. So this is our first step. This is our second step. This is our third step. This is our fourth step, fifth step, sixth step, seventh step, and this is our eighth step. So that is our eighth step. So we've realized that dehydrogenation occurs at what steps? That is step 3, 4. So we are seeing step 3, step 4. Another dehydrogenation is at step number 6 and step number 8. So dehydrogenation normally involves two types of coenzymes in the TCA cycle, that is NAD and FAD. So it involves NAD and FAD. FAD is not charged, but FAD is charged. So the first two steps involves NAD. The first two steps and the last two steps. The first two steps and the last step involve NAD. So step number 6 is the one that involves FAD. So that is what you are supposed to know. So we go back to our step number 3. So we are transferring hydrogen atoms from isocitrate onto NAD. So when we transfer those hydrogens, we convert our NAD into NADH plus a proton. So that is step number 3. Step number 4, we do the same thing. That is NAD into NADH. So step number 6, okay, you also need to know that NADH is always it takes the two protons, but it gets attached to one of the protons, and then the other one is normally free. That is NADH plus a hydride. That is a proton. Step number 6, we are using FAD. So FAD is converted into FADH2. Then step number 8 is a dehydrogenation reaction. So this one will involve the transfer of a hydride from a malate onto NAD again. So that is our NAD is converted into NADH plus a proton. Into a proton. So that is basically what we do to remember the core enzymes. So there is something else that we need also to remember. I promised you that I was going to explain the difference between a synthase and a synthetase. So synthase is self-explanatory. It is used to synthesize something. A synthetase involves synthesis of something, but it requires a nucleotide phosphate. So in this case, we need GTP, we need GDP. So GDP interacts with an inorganic phosphate to form GTP. That is the major difference between synthase and synthetase. Then another thing that you also need to remember is the first two of the dehydrogenation steps are accompanied by decarboxylation. So decarboxylation refers to the kicking off of carbon dioxide from a molecule. So we are going to kick off carbon dioxide from isocitrate to form alpha ketoglutarate. So these first two steps involve decarboxylation reactions. Then you also need to remember something here. You need to remember this step, the conversion of fumarate into malate. So these steps involve the addition of a water molecule across the double bond of fumarate. So here we are going to add water molecule across the double bond. So if you want to understand that, follow me on YouTube. In terms of biochemistry, I have described the TCA cycle in terms of chemistry. So that is basically what you are also supposed to remember. So let's go back to step number one. Step number one, oxaloacetate interacts with what we call water molecules and a 2-carbon component known as acetyl-CoA. So this is normally our first step, where we interact oxaloacetate with the water molecules and a 2-carbon component known as acetyl-CoA. So oxaloacetate interacts with water molecules with the acetyl-CoA in presence of water molecules to form citrate. So that is our first step. Second step is an isomerization reaction where we convert citrate into isocitrate in the presence of aconitase. So aconitase is a special type of isomerase enzyme that helps in the process. Then we go to the next step is a decarboxylation reaction. So we carry out the decarboxylation reaction. The purpose of isomerization is to rearrange the molecules for the forward reactions. So when we carry out decarboxylation of isocitrate into alpha ketoglutarate, we also carry out the dehydrogenation reaction. So the next step is facilitated by alpha ketoglutarate dehydrogenase. The same process repeats where we are dislodging hydrides from alpha ketoglutarate and adding them onto NAD to form NADH and kicking off carbon dioxide so that we form succinyl-CoA. So the next step requires a GTP. So that is conversion of succinyl-CoA into succinate, and that is succinyl-CoA synthetase enzyme facilitated reaction. So followed by succinate being converted into fumarate. So we meet this enzyme in the electron transport chain known as complex 2. So in electron transport chain, we refer succinate dehydrogenase as complex 2. And the purpose of succinate dehydrogenase is to carry out dehydrogenation of succinate into fumarate. So the transfer of 2 hydrogen atoms from succinate, that is the middle carbons of succinate, onto FAD to form FADH and fumarate. So fumarate is a double bonded molecule. So the next step is followed by addition of a water molecule across the bond to form malate. Then malate is converted into oxaloacetate. That is how you can easily remember the TCA cycle. TCA cycle in your exams. So we go to the next step. The next step says explain the meaning and benefit of the following in medicine. So the first thing that is we want to define here is empiric therapy. We want to know the meaning and the benefit of empiric therapy. So empiric therapy refers to early intervention through the use of antibiotics before a pathogen is identified. So physicians use this information from history and physical examination and any other complicated diagnosis to determine the antibiotics to use. Then we have another term known as antimicrobial prophylaxis. So this refers to the use of antibiotics to treat diseases, e.g. prevention of TB among individuals who are in close contact with infected patients. Also, pre-treatment of patients with artificial heart valves who are undergoing dental procedures. So stay tuned so that we go to the next question. Thank you so much. And don't forget to like, share, and subscribe.